Welcome all of you. Welcome students. Am I loud and clear to all of you? Are you getting my voice? Please reply in comments. My voice is reachable to you, all of you. Okay. So in the today's lecture, we are going to start with the topic that is explanation of the practical, that is uh, agarose gel electrophoresis, or we are going to see actually in details about the plasmid DNA isolation and genomic DNA isolation. Okay. So let's begin with the topic. As I have discussed with you that the topic for today is DNA isolation. So we will be beginning with it. Whatever the diagrams that I am drawing on the board or whatever the information I am giving on the board, you can take a screenshot of that. So let's begin. In the DNA isolation or genomic DNA isolation, one thing that you should know is the nature of genetic material. Okay. So today's topic is plasmid and genomic DNA isolation from bacteria. Okay, so the organism that we are using here is bacteria. So from bacteria, how you can isolate the plasmid and genomic DNA here in this in today's lecture. Okay, so if there is any question, you can comment here regarding the explanation. So we shall be beginning with the explanation. First of all, let us discuss the nature of genetic material. Okay, so there are two genetic materials that we are discussing here, or regularly there is a presence of genomic DNA in bacteria, and the secondary material or the extra chromosomal material that is present in bacteria is the plasmid DNA. Okay, so generally the plasmid DNA here, isolation of plasmid DNA is generally the major experiment in various syllabuses, and genomic DNA isolation from bacteria is the minor experiment in various syllabus of different universities of molecular biology. Okay, so you know the nature of the genetic material. I think here the genetic material may it be a plasmid DNA or genomic charge. Yes, and there are three most important entities is present in DNA. So these three entities that are present in DNA are, first of all, it's phosphate group, phosphate group. Then there is the presence of ribose sugar, and then there is the presence of nitrogenous bases. Okay, so nitrogenous bases can be A, T, G, C, and U in case of RNA in place of T. Okay, so these are the different nitrogenous bases that are present in DNA. Okay, so the major physical property of the DNA that is charged is due to the presence of phosphate group. That is, the negative charge is present on DNA on the basis of the phosphate group present, that is present in the DNA. Okay, so we are using the chemical properties of DNA in order to isolate the DNA from a mixture of samples or a cellular mixture. Okay, so naturally a cell is a mixture of different biomolecules. You can tell the cell as a mixture of different biological molecules. Okay, so how we can isolation of genomic DNA or plasmid DNA here means the purification of the DNA only from a group of biological molecules such as proteins, carbohydrates, lipids, then RNA, etc. Okay, so you have to isolate the genetic material, that is a genomic DNA or a plasmid DNA, from bacteria, from all other biomolecules that are present in cell. Okay, so we are going to see the whole process of this in details. So the whole process can be divided into three major types. You first write down these three entities and the nature on which the separation of DNA is based. Okay, so let's see what are the different criteria that helps in purification of DNA. First of all, we will be beginning with the different characters, genetic characters that are present in the DNA. So let's start with the purification process. So what happens when you are going to isolate a genomic DNA? So the isolation of genomic DNA from bacteria is a easy part. Why? Genome is a much complex genome. Okay. So the organization of the DNA in eukaryotic genome is a complex one, and it is there are different nucleosomes that are present in eukaryotic genome. But in bacteria, it's a simple nucleoid. It is a simple nucleoid form of DNA. Okay, so the nucleoid can be a central protein in which the DNA is entangled like this in case of bacteria. So and here there is a presence of number of histone proteins that helps in interaction of the DNA. Okay, so there are different histone like proteins that are present in bacteria also that helps in adhering the DNA to the nucleoid. Okay, so since the complexity is much lower as compared to the eukaryotic cell, the isolation of DNA from bacteria can be done easily. Okay, so there are different methods, or there are different combination of three different techniques that we can use here for the isolation of DNA, genomic DNA from bacteria. See, remember, plasmid DNA is also a kind of DNA. Okay, so the starting procedure for isolation of genomic DNA as well as plasmid DNA is same. Okay, so first of all, we are going to see the common isolation of DNA technique, and then we will be differentiating between the genomic DNA and plasmid DNA. Okay, so as I have told you, there are three different techniques that helps in isolation of DNA. The first technique is differential solubility technique or property, you can call it as. First is differential solubility. Second is adsorption method, and third is density gradient. Density gradient helps in isolation of DNA. Generally, in laboratory protocols in molecular biology, we utilize the differential solubility or the differential solubility of DNA with respect to other molecules for the isolation of DNA. Okay, you can use adsorption method in which the DNA is binded to a solid support such as silica column based method, where the DNA binds to the silica and all other biomolecules are removed from the mixture. And then density gradient centrifugation, that is CSCl2, that is cesium chloride density gradient centrifugation, in which the DNA forms a band in a centrifuge tube level. So it depends in that centrifuge tube in the density gradient centrifugation. This kind of centrifugation we have seen in the experiments of DNA, where we have determined the semi-conservative nature of the DNA replication. Okay, so in that experiment we have seen the centrifugation, that is cesium chloride density gradient centrifugation. Okay, so these are the different techniques techniques or types that can be utilized in the isolation of DNA. Generally, the lab protocol involves the differential solubility that we are going to see in details here now. Okay, so the steps in the isolation of DNA. So one by one, we are going to see all the three steps. The first step that is very important in the isolation of DNA is the cell lysis. Okay, so you know that the DNA is present inside the cell. Okay, so for its isolation, the cell has to be bursted out. Okay, so for the bursting of cell, you require cell lysis. Okay, so the first and foremost step in the isolation of DNA, whether it is a plasmid DNA or a genomic DNA, is the cell lysis. Okay, the second step that is present is the differential solubility or phase separation. Okay, differential solubility, or it is also called as a phase separation. Okay, then the third step in the DNA isolation here is the precipitation. Okay, so these are the different techniques or the different steps, that are three major steps that are involved in the isolation of DNA. So let's see the first step in details, that is cell lysis. First of all, there are different reagents, chemicals that can be used for the lysis of the cell. So first step that is there is the cell lysis. So what happens in cell lysis and how we can do the cell lysis, that we are going to see. The cell lysis procedure is involved in cell biology also, means there are practicals in cell bio also where the cell lysis takes place. So in this cell lysis, what is done, there are different chemicals or different enzymes that are used for the lysis of the cell. Okay, so lysis of bacterial cells specifically, because we are seeing the isolation of plasmid DNA and bacterial DNA in bacteria. Okay, so cell lysis can be done using two counters. The first by which you can lyse the cell is the chemical method, and the second is enzymatic. So in chemical methods, there are different chemicals that are used for the lysis of the cell. So there are some chemicals such as SDS. SDS is sodium dodecyl sulfate. Then there is C-tab. Then it can be a hot alkali such as NaOH, for example, NaOH. Hot alkali can also be used. Then there are DNA stabilizing buffers such as Tris HCl, EDTA, etc. So these are the different chemicals that are used for these cell lysis. These chemicals, what they will do, when you subject the cell to these chemicals, they are going to lyse the cell, they are going to break the cell, and when the content of the cell is released, the DNA present in that cell is also released. Okay, so now that DNA has to be stabilized, so it is done by Tris HCl and EDTA. It maintains the pH of the DNA as well as it maintains the structural integrity of the DNA, because we want the DNA in a proper form. Okay, so these are the different chemicals that are present in the cell lysis. So enzymes are used for the disruption of the cell or cell lysis. So let's see the different enzymes. In case of bacteria, when you are going for the isolation of DNA in bacteria, the main enzyme used here is lysozyme. Then proteases is used for the degradation of protein. For the degradation of RNA, RNase enzyme is used. And if the organism is different, so for suppose you are using a different microorganism such as fungus, then chitinase enzyme is used. It depends on the cell wall composition. Lysosome is help in breaking the cell wall of bacteria. So here for the bacterial isolation, the lysosome is used, lysozyme. Then proteases and RNase for degradation of protein and RNA, and chitinase in case of fungus. And if you are using a plant material, then cellulase enzyme is used. Okay, so such kind of enzymatic treatment also helps in cell lysis. Okay, so this is the first step in which the cell lysis is done, and all the content of the cell is released outside, and the DNA is also released outside. The DNA present in that is stabilized by using special salts such as Tris HCl and EDTA, which maintains the pH of the DNA also, and helps in protection of the DNA from sharing from these chemicals. Okay, there may be other chemicals also in some different methods, but the final aim of these chemicals is same, that is the breaking of cell wall or cell membrane in order to release all the cellular content. Okay, so let's see the next step. First, now you have seen here the cell lysis process. Now the cell has lysed here. The next step that we are going to see here is the differential solubility. Okay, the next step is differential solubility, or it is also called as a, in this, see, it is based on the DNA solubilities in a particular organic solvent. Okay, so generally DNA is solubilized in different organic solvents. So depending on the solubility of DNA in these solvents, this second step is there, that is called phase separation. Okay, so generally the solvent that is used in solution of or the solubility of DNA here is phenol isoamyl alcohol. Okay, so generally these are the different, these are the two solvents. There may be another solvents also that helps in differential solubility of DNA, remember, okay? And then there is another chemical that is also used sometimes is chloroform. Okay, this does not solubilizes the DNA but helps in phase separation. Okay, so what happens? Whatever the cell that has been lysed in the upper step. So in this step prior to this, that is the cell lysis, you have busted the whole cell. You have released the content of the whole cell. Now what you are going to do? You are going to take the all the components of the cell into the tube. For example, suppose this is the Eppendorf tube. That is a special glass-made tubes that are used. These are called as vials for the DNA isolation because you know DNA has a property of adhering to silica. Okay, silica means glass. Okay, so DNA property of binding to glass. Okay, so in order to prevent that, you cannot use glass tubes for the DNA isolation. So plastic tubes are used. That is called as a Eppendorf tubes, and it is simply called as a vials. Okay, so these are available in 1.5 ml, 2 ml, then zero point 0.5 ml, then zero point 0.2 ml capacity. Okay, like these capacities are there. So these are available in these capacities, and these specialized tubes are used for DNA isolation. Okay, now see, for example, you have now taken the solution of bacteria which is suspended in the first solution, that is cell lysis solution. Okay, so due to the cell lysis solution, what is going to happen? All the DNA that is being lysed here, or the cells that are being lysed is present in this solution. Okay, so after this equal amount of you are going to add equal amount of the solution second, that is the differential solubility or phase separation solution, in which it can have fluoroform or chloroform isoamyl alcohol. The either of the two solvents, that is phenol or isoamyl alcohol, will be taken here, and it will be taken along with the chloroform. Okay, so there is, see, depend on the bacterial concentration or the bacterial DNA, the ratio of phenol chloroform can be 1 is to 2, or it can be 20 is to 1. If you're using isoamyl alcohol, then it can be 20 is to 5. So such kind of different ratios can be taken for the phenol chloroform or chloroform isoamyl alcohol mixture. Okay, it depends on the DNA that you are going to isolate. Okay, so equal amount of that solution you have to add here. So for example, I am adding the phenol chloroform solution. Phenol chloroform solution 1 is to 20. Okay, so what happens here? After adding this, the tubes are mixed by inversion. Okay, you have to mix the tube like this, to and fro motion. You have to hold this tube and mix it like this, so that the whole solution gets mixed. Okay, so due to this, what is going to happen? The cellular extract or the cell lysate is going to mix with the phase separation solution here, and then you are going to centrifuge or spill these tubes at high speed. Okay, so when you spin these tubes at high speed, what is going to happen? There will be a phase separation here. Okay, I'll show you the diagram how the phase separation generally occurs here. You can see this is the Eppendorf tube again. I'll draw it another color. Suppose this is the tube here. So now what happens? The layer is like this. Okay, right at the middle of this, there are three phases that are formed here. Suppose you are using the phenol chloroform, there are three different layers that are formed here. In the upper layer, see, this is the upper layer, and there is a layer. Okay, okay, so what happens here in this, the layer below, there are different extract, cellular extract in the layer below. Okay, and this middle layer is the proteins and cellular debris. Okay, so the middle layer will have all the proteins present in the cell after centrifugation. After the centrifugation, you are going to get these uh, different layers. Okay, so the middle layer, that is a thick layer or whitish colored layer, it is the proteins and cellular debris that gets accumulated in the center, and the last layer in the bottom of the tube is the cellular extract or cell solution, and the upper phase is the phenol. Okay, in phenol there is a DNA that is solubilized. Okay, so all kind of DNA will be there here, all kind of nucleic acid. Actually it will be genomic DNA as well as plasmid DNA as well as RNA. Okay, so here in this phenol phase there will be DNA, RNA, plasmid. Okay, all kinds of nuclei will be present in the phenol phase, so therefore it is called as a differential solubility or phase separation. Okay, so what happens in this, you get a different phase of the DNA or the nucleic acid present in the cell, that is DNA, RNA, plasmid, in a separate fraction that is called phenol fraction. Okay, so therefore it is differentially solubilized here. Okay, so let's see after this what happens. So after this, what is going to happen? You are going to take the upper phase. Okay. So you are going to take a new tube here. Suppose this is the new Eppendorf tube, and you are going to transfer the upper phase only. The phase having the DNA. Okay. So suppose this is the upper phase here. What you have done, you have just separated the upper phase carefully using a micropipette. Okay, you have used a micropipette here, and carefully, without disturbing the middle portion or the middle layer, you are carefully taking out the upper phase into a new tube. Okay, so what this phase will have, this phase will have all kind of nucleic acids such as DNA, RNA, and plasmid. Okay, so when you separate this, you are going to use this tube for the next step, that is the precipitation. Okay, so let's see the next step here, that is the precipitation. The next step is precipitation, that is the third step. Okay, precipitation. Okay, so what happens in this step, I'll tell you. In this step the DNA is precipitated. Okay, so the DNA is precipitated. How the DNA is precipitated? So normally what happens, DNA is solubilized. Okay, normally what happens, DNA is solubilized in the organic solvent. Okay, so in this step what are you going to add organic solvent such as phenol. Okay, in the upper phase. Okay, so now here is precipitation solution. Okay, the precipitation solution is generally isopropyl alcohol or ethanol. Okay, so these are the two different solutions that are used as a precipitation solution. You can take this solution as 100%, but the main thing is you have to take this solution in a chilled form. It should be absolutely cold solution. So when you add this solution, what happens, in the earlier step I have shown you, here there is a uh, upper phase that you have collected in the previous step. Suppose this is the upper phase, and you are going to add a solution that is chilled isopropanol or ethanol to this. Okay, so what happens here, see, I'll show you here. Suppose this is the tube again, and this is the phase that was there, and you have now added a chilled isopropanol or ethanol solution. Okay, so this is the isopropanol that is shown in blue color here, the below is the green color. So now what is going to happen, you have to add this chilled isopropanol or ethanol here in order to precipitate the DNA. Okay, so what is done here, due to the presence of DNA, due to the presence of this alcohol or isopropanol, what happens, this tube is then mixed by intermix. Okay? You have to mix the tube by to and from motion. So when you mix the tube, the whole solution gets mixed, and the phosphate bond or the phosphate bond that present in the DNA is neutralized here. Okay, so due to this, the DNA becomes hydrophobic and starts precipitating. Okay, so in the next step, when you mix this properly, what is going to happen, you have to keep for 1 hour, or depending on the concentration, it can be a whole day also, or 12 hours also. So you have to keep this solution, and then what is going to happen in this solution, all the solution will be like this, mixed like this. Okay, and the total DNA that is present will be precipitated out. Okay, so DNA will be precipitated out in the bottom of the tube like this. Okay, and due to this is due to the neutralizing of the phosphate bond that is present in the DNA, and due to this, what is going to happen, the DNA become hydrophobic in nature. Hydrophobic nature of the DNA in the solution, it gets separated out of in the solution and gets precipitated in the bottom of the tube. Okay, so now this DNA can be purified further by using centrifugation. Okay, when you go and centrifuge this tube, what is going to happen, this DNA, all the DNA being precipitated in the whole tube is going to stand in the bottom. Okay, it is going to stop in the bottom. Okay, and then what you can do after centrifugation, you can remove the supernatant by decanting. So when you remove the supernatant by decanting, when the DNA that will be present in the solution is going to precipitate out, and then you can keep the tube as it is. Okay, so suppose centrifugation is done now, and now fully here, after the addition of, how you can distinguish between the plasmid and then RNA and the genomic DNA, what happens here, if you are going to isolate the genomic DNA, you are going to proceed like this only. Okay, just like I have told you, you are going to precipitate the DNA, and then after that you are going to purify the DNA formed, and that pellet is added to the DNA solubilizing buffer. So in that, then you are going to treat with the different RNase enzyme to remove the RNA contamination, and then you get the purified DNA. Okay, but when you are going to isolate the plasmid, remember, you have to isolate the plasmid DNA of genomic DNA. Then here you are going to add one more solution, that is potassium acetate. You add two different solutions, or you can add any one of them. Okay, you can add acetate or sodium acetate. The pH of solution should be from 5 to 5.5. Okay, due to this, what happens, these two solutions, that is potassium acetate and sodium acetate, are also called as a renaturing solution. But if you don't add, you are going to get the genomic DNA. But when you are going for the isolation of plasmid DNA, specifically, you have to add sodium acetate and potassium acetate solution. The function of these solution fastens the rate of renaturation. DNA renaturation rate is fastened by this, or the double helical DNA formation rate is fastened by this. Okay, so what happens, since the plasmid molecule is a very small molecule and the genomic DNA molecule is a way larger molecule than the plasmid. Okay, so what happens, that when you are going to treat the cell with the cell lysis solution, DNA gets denatured, means DNA form comes into the single stranded form. Okay, so when you are precipitating, DNA again renaturates and comes to its double stranded form. Okay, so normally this happens over a period of time. Okay, so the process becomes faster. Okay, so due to this, the renaturation of the DNA gets faster. Here the difference lies that when you add the renaturation solution, the plasmid DNA that is being smaller renaturates faster. Okay, plasmid DNA being smaller renaturates faster. Okay, so due to this, then within 5 minutes of the addition of potassium acetate and sodium acetate solution, you are going for centrifugation. Immediately after that, you are going for centrifugation. Means what? whatever the genomic DNA is present in the solution, you are not giving time for that genomic DNA to get renatured in the plasmid within the 5 to 10 minutes in. Okay, and then after that you are going to go for centrifugation. uh, so due to this, what is going to happen, the plasmid DNA is only going to form a precipitate because you have given time only for plasmid DNA to get renatured. Okay, and then only plasmid is able to form the precipitate in such a small interval of time, and when you centrifuge that, the plasmid is going to form the pellet immediately, and the genomic DNA is separated in this, which takes a longer time for renaturation. Okay, thereby you are separating the plasmid in the pellet form, and you have to then remove the supernatant, discard the super, then you have isolated the plasmid here like this, and you can isolate the genomic DNA also. If you give a longer time to this, what is going to happen, the genomic DNA is also going to form the whole pellet in the bottom of the tube. Okay, so thereby you can get a genomic DNA and a plasmid DNA, in the solution separately. Okay, so after this step, after the purification of this plasmid or genomic DNA, it depends on the way, or it depends on what do you want. You want genomic DNA or plasmid DNA? It depends on that. What are you going to purify? So in the next step, what will happen? You are going to rinse the DNA. Okay, after the precipitation, you are going to rinse the DNA. Okay, suppose you have isolated both plasmid and genomic DNA. Okay, some tubes. So this is the first tube, this is the second tube. In the first tube, you have, sometimes you are not able to see this plasmid DNA. It is so less in amount, or very light, very low amount of plasmid is there in the cell as compared to the genomic DNA. Okay, this is for plasmid and this is for genomic DNA. Okay, so you are going to add it with a buffer, because you have to purify the DNA also. See, proteins and other debris are being removed, but there is a presence of RNA along with this. Okay, you have RNA contamination along with this, because RNA is also a nucleic acid. So naturally, RNA will also come along with this in the plasmid DNA and genomic DNA precipitate. Okay, so both the tubes have to be added with the RNase enzyme. 3 microliter, about 3 to 5 microliter treatment is there, where the RNase enzyme, that is RNA degrading enzyme, is added into this. The RNA present in these solutions will be degraded. Okay, and you get a purified DNA. Again, after the treatment of RNase enzyme, what are you going to do? You are going to go for the ethanol rinsing. Ethanol rinse with 70% concentration. Okay, so you are going to add 70% ethanol to both the solutions, and again going to rinse this solution. So this tube separately is precipitated. Okay, and the degraded RNA is present in this buffer here. Okay, so it will have degraded RNA here in the upper supernatant, and that supernatant can be discarded and the pellet can further analysis. Okay, so thereby you are getting the plasmid and genomic DNA in a purified form, and in a very much purified form. Okay, so how you can check whether the plasmid or the genomic DNA that you have, you can check out that video from my channel, purification of DNA. Okay, how you can analyze whether a DNA is purified or not. You can do that by taking the ratio of absorbance of at 260 nanometer by absorbance of that DNA at 280 nanometer. Okay, so when you do this, you get a ratio. Okay, absorbance at 260 nanometer and absorbance at 280 nanometer. Okay, 1.78 actually. Your DNA is 100% pure. Okay, so once you analyze the concentration of the DNA using this formula, then you can go for the analysis of the DNA using different methods. Okay, so there are two different methods of the analysis. Okay, it depends on what kind of analysis you want to do. Okay, Suppose if you are qualitatively analyzing DNA, that is only the DNA is present or not, then go for DPA test. Okay, DPA is tube and just you reaction, and then DNA is going to form a blue color. Okay, so get confirmed that the DNA is present here. Okay, so this is the qualitative test. But if you want to amplify or analyze the DNA quantitatively, that is, you want to know its molecular weight, you want to know its charge, and you want to separate it based on the size, then you have to go for the quantitative analysis, that is the agarose gel electrophoresis. Okay, so the quantitative analysis can be done using the in the analysis, that is the agarose gel electrophoresis. It is agarose gel electrophoresis. Okay, so let me first, the agarose gel electrophoresis involves the separation of the DNA on the agarose gel. Okay, so first of all, let's see what is agarose. Agarose, it's a polysaccharide. It is a polysaccharide. Remember, or it can be called as a polymer also. Made up of the repeating units of disaccharide, that is called as agarobioes. Okay, agarobioes. This is a disaccharide that is repeated monosaccharide. Okay, one is D-galactose and anhydrogalactose. Okay, so these are the two different forms of gel that is isolated from a seaweed. This weed is present in sea, that is the red algae, that is from the genus Gelidium. Okay, so from the genus Gelidium, there are different species of Gelidium. This red algae from which this agarose is isolated, and then it can be used as a used in agarose gel electrophoresis. Okay, so let me first draw the structure of agarose. That is structure. Show you the structure of agarose here first. Okay, so I was drawing the structure of agarose here. The connecting link this side. Okay, remember. So it is shown in bracket here. You can see I've shown the bracket here. This whole disaccharide unit is agarobioes, that is being repeated over and over. Here there is the presence of H. Here there is oxygen connected with this oxygen, that is third and sixth position. Therefore, it is called 3,6 anhydrogalactose structure. Okay, so this is the structure of 3,6 anhydrogalactose. Both these structures, that is a disaccharide, D-galactose and connects together and forms agarobio. Over and over again, you can see the connecting link here, and this connecting link continues over here. Okay, so this continues, and this whole polymers is formed, and then the polybios. Okay, so now when the agarobioes is formed, this agarobioes is used for the formation of agarose gel. Okay, I hope you have seen the structure of agarose now. So now let me tell you how this gel is formed and where you can separate the isolated plasmid DNA or genomic DNA. Okay, remember you have to prepare the agarose gel in a specific buffer that is called as a TAE buffer. Okay, the buffer is called as TAE buffer. The full form of TAE is tris acetate. Okay, the EDTA full form tetra acetic acid. Okay, ethylene diamine tetraacetic acid. Okay, so here this TAE buffer is prepared. You have to generally use the 1x TAE buffer. Generally 1x TAE buffer is taken and you have to add it in water, and this in 1x TAE buffer for the visualization. Normally what happens, DNA is not visible, so for the visualization purpose 10 microliter of 100 mg per ml ETBR is added. ETBR means ethidium bromide. This is added in this medium. So this helps in visualization of the DNA into the agarose gel. Okay. So generally the agarose gel that is prepared is 1% for regular genomic DNA samples. It can be taken as 0.8% for plasmid DNA. Since the plasmid DNA is very smaller in size, you take the concentration of agarose in a smaller amount. Okay. So when you add this in a beaker, you are going to add 1%. For example, 50 ml agarose gel I am preparing. 1x TAE buffer, 1x TAE buffer 50 ml you have to add, and in that you have to add 10 microliter of ETBR. Okay, okay, and you have to pour it on a agarose gelling plate. Okay, so when you pour it on the agarose gelling plate, you have to form the combs here or wells here in which the DNA will be uh, loaded, or the sample will be loaded. Okay, so I will show you a structure of a rough gel. For example, suppose this is the gel that is being formed here. This is the gel that is formed. So these are the different wells that are being formed from the agarose gel. Okay, and just this is for just for demonstration purpose. So you can put the DNA sample in this well, a plasmid genomic DNA sample, or it can be a plasmid DNA sample. Okay, and then you have to put this gel into a agarose gel electrophoresis apparatus or assembly. Here there will be positive side, here there will be negative side. Here, the well side is always negative, sorry, and the other side is always positive. Okay, so due to this, what is going to happen when you load the DNA onto the well here, the DNA will be moving towards the positive side. Since the DNA is negatively charged, DNA moves to the positive side, and thereby the separation of DNA takes place. And from the separated DNA, you can analyze the DNA's molecular weight. You can put a DNA ladder onto this side. So DNA ladder have the different bands of different molecular weight, known molecular weight. So by comparing, suppose this is the band that you are going to form. Okay, suppose this is the genomic DNA and it is forming a single band here, genomic only one. So it will correspond to a particular bond in the DNA ladder, and you know the different molecular weight of the DNA ladder bands. Okay, and then you can tell the molecular weight of this particular band using the comparison or the comparing it with the DNA ladder. Okay, similarly you can do it for the plasmid DNA also. Okay, so this was about whole process of explanation of the genomic DNA and plasmid DNA isolation. The in the agarose gel electrophoresis is dependent few points. The first is the agarose concentration. How the DNA is going to separate on the agarose gel, it depends on the agarose concentration, first point. Then second point, it depends on the conformation of the DNA. Whether it is single stranded DNA or a double stranded DNA. Single stranded DNA generally moves fast, stranded DNA moves slower. Okay. Then it also depends on the voltage that is applied to the agarose gel. Okay, so now generally the voltage is 50 volts to 100 volts. If you are using higher voltage, the gel may melt here, so therefore the electron done between 50 to 100 watts, and it generally takes 1 to 1.5 hours for proper electrophoresis to take place. Okay, so along with the ETBR, in the DNA sample, in the solution, it is just the solution that you are able to see. Therefore, for the visualization of the DNA or visualization of the solution, you add two dyes. One is xylene, cyanol, and second is bromophenol blue. Okay, so these two dyes you are going to use so that you can track the DNA here in the solution. So this xylene, cyanol, and bromophenol blue, these are called as a tracking dye. What it is called? These are called as a tracking dye that helps in tracking of DNA into the agarose solution. So when the agarose gel, the dye reaches on this edge of the gel after the electrophoresis, then you have to put off the current so that the DNA does not cross the agarose gel and comes out of the gel. Okay. So therefore, it helps in tracking of DNA. Therefore, it's called tracking. There are two different dyes that we use here. One is xylene phenol, and second is bromophenol blue. Okay. So this was all about the details of the isolation of plasmid DNA and genomic DNA. I hope you have understood the everything in details. If there is any question, put your question in comments. Thank you for today's video lecture. Thank you so much. I hope you have understood this.